Hello, I'm John Nichols, and for 42 years, I was an English GP or family doctor in my hometown of Guildford, which is quite near London. Now, one of the good things about Guildford is we have our own university, University of Surrey. And during my career as a GP, I collaborated with colleagues at the university on health promotion and research projects, including those to do with the gut. And so today I have a question for you, which I'm going to bring up. And here it comes. Are probiotic bacteria really our little friends? Are they our friends or what? Probiotics are a bit controversial generally, so let's have a closer look. Well, probiotics are symbiotic bacterial organisms that we do better off if we have the right probiotics in our microbiota or gut flora, as we used to call it. And they have these qualities. Well, first of all, a symbiotic organism or symbiotic partnership is living together of two unlike organisms. And a human being and a bacterium are certainly poles apart on the tree of creation. Symbiotic organisms are basically opportunistic rather than altruistic, as we shall see. A symbiotic partner can turn against you if circumstances change. On the other hand, altruism is, dictionary definition, behavior that is unselfish and may even be detrimental to the individual. It's mainly seen in humans, but also in other animals, particularly a mother will be hostile when she has young and will do just about anything to protect her young, and maybe even risk her life. Altruistic behavior in herds of animals and in human society is usually biased towards genetic inheritors. Therefore, as well as parents being protective of their children, grandparents will also sacrifice themselves in various ways to protect their children. Also, we might be quite protective of nephews and nieces, and even slightly more distant relatives, but very soon it fades out. So altruism is certainly got a genetic basis. And true interspecies altruism is most unlikely. I'm not saying it never happens. Think of the loyalty of a dog to his master or mistress. That maybe is getting in that direction, shall we say. And here is the classic schoolroom example of a symbiotic partnership. The hermit crab, after it's found its shell, is likely to look for a healthy looking sea anemone and pluck it off its rock and plonk it on its shell. And this is a mutually advantageous relationship. It is a symbiotic relationship because if the crab comes across a tasty morsel, a small dead fish, for instance, it can shred it up with its claws, and little bits of this snack will float up to the sea anemone, and the sea anemone will have a nice snack too. But the other way around, it works too, because the sea anemone's tentacles have stings that help to protect the hermit crab against parasites and hostile other animals. But sometimes a hermit crab can get very hungry. If you were to put it in an aquarium all on its own with nothing to eat, it would get very hungry. and then it may well reach back over its shell, pluck off the sea anemone, and eat it. So that can happen. So trans-species altruism is almost unknown and plays no part in the average symbiotic relationship, or as the hermit crab might well comment if he could, there is no place for sentimentality in this relationship. So what drives a successful symbiotic partnership, such as the symbiotic partnership between our gut microbiota and ourselves? Well, the important thing is co-evolution. The two partners evolve to get closer and closer and more dependent upon each other over perhaps millions of years. In fact, it might start off as a relationship between a host and a parasite and gradually evolve into a symbiotic partnership. And here's a good example of co-evolution. Over a relatively short period of just 50,000 years, there's been a symbiotic relationship developed between man and wolf, evolving into dog. So co-evolution is highly relevant to the evolutionary process and may explain why Homo sapiens prevailed over Homo sapiens neanderthalis, because Neanderthal man did not have dogs. So when Homo sapiens sapiens, that's us, started to move into Europe, for instance, we had our hunting dogs, which gave us a big advantage over the Neanderthals. So let's look at what happens. First of all, over 50,000 years ago, when the wolves howled on the hillside, we huddled close around our fire, hoping that they wouldn't come down and steal one of our children. But some wolves became camp followers. They would survive on scraps thrown out by humans. Maybe some humans are a little more generous than others, but certainly old bones thrown out would be a little snack for a hungry wolf. But this relationship got closer, and some clever humans adopted wolves and used them to help in their hunting, as you see here. And this became a closer and closer evolution of a relationship, became closer and closer and more efficient as we became better hunters and the dogs became better hunters' assistants. And finally, it has evolved into a relationship between an old man and his dog that takes him for a walk, which is still a beneficial symbiotic relationship, I'm sure you'll agree. Here's another example which involves this gene for virulence. As you can see, the common streptococcal ancestor to two varieties of this streptococcus has 3 of these genes. So what do we mean by genes for virulence? Well, they may work in various ways, but one of the easiest to understand is that it makes the organism a more effective invader of its human or mammal host, because it forces the immune response to become very exaggerated so that the inflammatory mediators go wild and cause tissue damage. And in this damaged tissue, the organism can spread more easily. But over 20 million years now, a slow process of evolution occurs, splitting into 2 streptococci, one of which is a friend and one of which is a foe. And if you look closely, you'll see that to begin with, it's lost one of its virulence genes on this evolutionary branch. And by now it's lost all 3 of them. And this is Streptococcus thermophilus, which is a friend, whereas Streptococcus pyogenes still got its virulence genes, and this is an enemy that could in fact kill you. Streptococcus thermophilus is a probiotic organism, which is found in dairy products, found naturally in dairy products. it survives well in them and is beneficial to human health, and is certainly found in many dairy products. So we have also a useful scientific word for the little friends that live in our gut, like Streptococcus thermophilus and various forms of acidophilus. And there's endosymbionts. Oops, here come two. It could be a symbiotic E. coli swimming past. And there they go, swimming off screen. And oh, here comes an enterococcus, a symbiotic enterococcus building away there. Now, here's another example of a symbiotic relationship, which is in some ways very similar. I went out into my garden and took a photograph of lichen. This is lichen on my mulberry tree. And here is lichen growing on my patio, a bit unsightly. They're completely flat, you can't trip over them, and they're very difficult to scrub off, as I've discovered. So what's happening in this symbiotic relationship? There are two unlike organisms. The green are microscopic algae, and they survive within a protective body of a fungus, which is represented by blue. And the algae are busy photosynthesizing away, creating sugar and energy to the benefit of both organisms. And they are protected within this fungal body, which puts out little roots called hyphae that anchor it to my patio, which is why it's so difficult to scrub off. So the mammal gut is full of these endosymbionts that are as beneficial to us as the microscopic algae are to lichen. Now, the symbiotic partnership between algae and lichen fungi has been extremely successful. You can find lichen growing quite prolifically from the jungles of the equator right up into the poles, in either poles. But what happens when they want to reproduce? Well, it's sexual reproduction, which is the most effective long-term strategy, only really applies to the fungus. And it produces spores, which when they land on something, and this spore has landed on a tree trunk here, they put out these rooty things called hyphae, and they're desperately searching, they're looking everywhere for the symbiotic algae they need to survive, and to persuade them into this symbiotic relationship. If they do not find their algae, and there's a limited number of species that will form this relationship, if they do not find that particular type of algae, they will wither and die. But humans are very similar, especially human babies, because this is why babies keep putting things in their mouth, because they're trying to top up their endosymbionts. Oh, you've got something green and yucky there, and now he's going off into the next room. I wonder what he'll find in the next room. But mother, don't be too keen to take the things out of his mouth when he puts strange things in his mouth, because he needs to do this to top up his endosymbionts in his gut to make his whole metabolism more efficient, as we shall see. Here is an example of what's going on in the gut. And this is not, this is a photomicrograph of a nodule lining the gut. It's not a cross-section of the gut. This is a nodule in the gut, which has got a hollow center. That's the hollow center. And here are the immune cells, the lymphatic cells, these blue blobby things that are the powerhouse of the immune system in our body. This is the gut-associated lymphatic tissue, or GALT. And 70% of our immune system is GALT. 70% of our immune system is in the lining of our gut, because what happens there is so important. And if you look carefully, you'll see the endosymbionts here, the little green and red spots, endosymbionts. And they are working for mutual advantage with the lymphatic cells. They are stimulating the lymphatic cells to do their job better, and they are having a safe home in return for this. So this is a useful symbiotic relationship to both partners. Research has shown that germ-free mice have a decreased secretion of intestinal antibodies, the IgA antibody response, and they have an immature structure of the gut-associated lymphatic tissue when compared with normal mice. They're less able to deal with various insults and injuries involving hostile microorganisms, for instance. And in humans, we have to be careful because newborn babies get the first good topping up of their endosymbionts through breastfeeding. So this is one reason why breastfeeding is better than bottle feeding. Also, they may even before that have topped up their endosymbionts in their gut microbiota during a normal birth, because as they go through the birth canal, they can suck up secretions from the vagina, from the birth canal, that include beneficial bacteria. So bottle feeding and a normal birth as opposed to a cesarean birth have been shown to both be beneficial for general health and in particular in relation to the gut microbiota. So what do we get out of our endosymbionts? Well, first of all, as I've already indicated, host protection against invading hostile microbes. This probably came about as a fortunate byproduct of natural selection on microbes to avoid them being replaced with other microbes. Secondly, the polysaccharides in our diet, which are part of dietary fiber, in the dietary fiber in vegetables, won't be digested by our digestive enzymes, but the gut microbiota can digest them and produce a substance called butyrate, which to the microorganisms is a waste product, but to the mammal host, it is liquid gold. It has some beneficial effects on the general health of the gut lining, and anything left over can be absorbed and used as a source of energy. In fact, it is so important a source of energy in some cultures, say an underprivileged child in a tribal area of Africa, the butyrate this child gets from digesting the polysaccharides could make the difference between starvation and good health. And thirdly, stabilization of the immune system. This started as evolved tolerance to endosymbionts and gradually moved into a more dependent relationship. We just know from research that the immune system works so much better when it is in tune with these endosymbionts. But as I said, our little friends or endosymbionts are opportunities that could turn on us at any time. So the question is, how do we keep them in check? They're microorganisms that could behave just like their ancestors as invading microorganisms, so they have to be kept in check. But I will list the ways in which we do that. But first of all, just consider what happens when we die. When we die, these ways of keeping the microorganism in check suddenly stop. And they have a field day. they start to invade our tissues and eat us. And that's why dead things are smelly, I'm afraid. It's a process called putrefaction. So here's how it works. The microbiome-host organism is a two-way traffic. There is an enormous metabolic impact from the gut microbiota on the human host, even to the extent of this metabolic impact affects the function of our brain, which is a long story, so I won't go into it just now, please. But the feedback and control is equally important, and this is how it works. There's a mucus layer and the lining of the gut, which is protective, and even it has some chemical components that selectively feed our friendly endosymbionts. These are glycans. Now, the lining of the gut, if it is being over-invaded by microbes, including these supposedly friendly microbes, can release oxygen that immediately kills off the invading microbes. Also, the gut lining produces a class of chemicals called defensins, which act like antibiotics. They're antimicrobial peptides chemically. Also, there's these immunoglobulin A, the IgA, that I mentioned earlier, that are produced as a result of the relationship between friendly bacteria and the gut-associated lymphatic tissue. These antibodies don't go into the blood, they're secreted into the gut, and they can be specifically aimed like magic arrow at specific organisms. As antibodies do, they're attuned to particular pathogens, such as salmonella. And here's a fifth way in which we keep the bacteria in check and control them. Our gut lining has another trick. It can produce little bits of microRNA. Now, RNA is like half of a DNA. So there's a DNA, the double helix. The RNA is a single helix produced off the DNA. And these microRNA snippets are able to penetrate the bacterial cell, and then they latch on to the bacterial DNA here. And they can modulate the bacterial DNA to do one of two things. If it's a microorganism that is beneficial and we want more of them, well, there you go. It can encourage them to reproduce. Or if it's a bad guy that we want to destroy, it can stimulate the organism to self-destruct, to go splat. So that's neat. But as I say, when we die, all these mechanisms stop. I owe a debt to Professor Kevin Foster of the University of Oxford, whose papers I've been reading, because many of these ideas come from him. He described the microbiota of the gut as an ecosystem on a leash. So it's a bit like our friend the dog that we have a symbiotic relationship with, but we keep him on a leash. And in the same way, these mechanisms I've described keep our endosymbionts, the microbiota, on a leash. So in summary, our little friends in our guts are opportunists that have to be kept on a leash, but we really couldn't manage without them after millions of years of evolution with them. If you want to learn more about the gut and the microbiota and nutritional medicine, you could try reading one of my books. Or you might like to look at my website here, which contains extracts from my books and my research. So that's all from me for now. So goodbye.